Carbohydrates, a review. Carbohydrates are a major class of biomolecules, essential for life and fundamental in various biological processes. They serve as a primary source of energy, provide structural support in plants and animals, and play crucial roles in cell recognition and signaling. This review covers the structure, classification, functions, and significance of carbohydrates. 1. Structure of carbohydrates. Carbohydrates are organic molecules composed of carbon, C, hydrogen, H, and oxygen, O, generally following the formula CH2ON. They are made up of monosaccharide units, which can be simple sugars like glucose or more complex structures when linked together. Each monosaccharide consists of a carbon chain with a hydroxyl group, NiOH, attached to each carbon except one, which typically forms a carbonyl group, either an aldehyde or ketone. 2. Classification of carbohydrates. Carbohydrates can be classified into four main categories based on their complexity. Monosaccharides, simplest form of carbohydrates consisting of a single sugar unit. Common examples include glucose, fructose, and galactose. They are often referred to as simple sugars and are absorbed directly into the bloodstream. Disaccharides, formed by the combination of two monosaccharide units through a glycosidic bond. Examples include sucrose, glucose fructose, lactose, glucose galactose, and maltose, glucose plus glucose. Disaccharides are broken down into monosaccharides during digestion. Oligosaccharides consist of 3 to 10 monosaccharide units, found in various foods and can act as prebiotics supporting gut health. Commonly found in vegetables like onions and legumes. Polysaccharides, composed of long chains of monosaccharide units, can be branched or unbranched. They are categorized into storage polysaccharides like starch and glycogen and structural polysaccharides like cellulose and chitin. Starch is the main storage form of energy in plants, while glycogen serves a similar function in animals. Cellulose provides structural support in plant cell walls. 3. Functions of carbohydrates. Carbohydrates play several crucial roles in biological systems. Energy source. Carbohydrates are the primary energy source for living organisms. Glucose, a monosaccharide, is metabolized in cells through glycolysis and the citric acid cycle, generating ATP, the cell's energy currency. During intense physical activity, glycogen stored in muscles is rapidly converted to glucose. Energy storage. In plants, energy is stored as starch, while animals store it as glycogen in the liver and muscles. These storage forms are broken down into glucose when energy is needed. Structural role. Cellulose is a key component of plant cell walls, providing structural strength. In animals, chitin forms the exoskeleton of arthropods and the cell walls of fungi. Cell recognition and signaling. Carbohydrates on cell surfaces play a role in cell-cell recognition and communication. Glycoproteins and glycolipids, which have carbohydrate chains attached to proteins or lipids, are crucial for immune system function and cellular communication. 4. Digestion and metabolism of carbohydrates. Digestion of carbohydrates begins in the mouth with the enzyme amylase, which breaks down starch into maltose. The process continues in the small intestine, where enzymes like maltase, sucrase, and lactase break down disaccharides into monosaccharides. Monosaccharides are absorbed into the bloodstream and transported to cells for energy production. Excess glucose is stored as glycogen in the liver and muscles or converted to fat for long-term energy storage. 5. Carbohydrates in diet and health. Carbohydrates are a significant part of a balanced diet, with whole grains, fruits, vegetables, and legumes being healthy sources. Complex carbohydrates, e.g., whole grains, vegetables, provide sustained energy and dietary fiber, supporting digestive health. Simple carbohydrates, e.g., sugars in sweets and soft drinks, can cause rapid spikes in blood sugar levels, which may contribute to conditions like insulin resistance and diabetes if consumed in excess. Dietary fiber, a form of carbohydrate that is not digested by humans, is vital for digestive health, reducing the risk of colon cancer and aiding in weight management. 6. Carbohydrates and disease. Diabetes mellitus, a condition characterized by impaired glucose metabolism, leading to high blood sugar levels. Management often involves monitoring carbohydrate intake. Obesity, excessive intake of refined carbohydrates can contribute to weight gain and obesity. Cardiovascular disease. Diets high in refined sugars and low in fiber may increase the risk of cardiovascular issues. 7. Emerging research in carbohydrate science. Advances in glycomics, the study of carbohydrates in biological systems, have shed light on their role in various diseases and immune functions. There is growing interest in the use of oligosaccharides as prebiotics to enhance gut microbiota and improve digestive health. The potential for carbohydrate based vaccines and therapies is being explored in the field of immunology and infectious diseases. 8. Conclusion. Carbohydrates are fundamental biomolecules that play diverse roles in energy provision, storage, and structural functions. They are essential for maintaining metabolic processes and supporting overall health. A balanced intake of carbohydrates, focusing on whole grains and fiber rich sources, is crucial for optimal well being. As research continues to unfold, the understanding of carbohydrates' roles in health and disease will deepen, offering new insights and therapeutic possibilities. This review highlights the importance of carbohydrates in both everyday life and scientific study, emphasizing their versatility and critical functions in biological systems. Monosaccharides, a review. Monosaccharides are the simplest form of carbohydrates and serve as the building blocks for more complex carbohydrates like disaccharides, oligosaccharides, and polysaccharides. They play a critical role in energy metabolism, cellular structure, and as precursors for various biochemical processes. This review provides an overview of their structure, types, biological roles, and importance in health and disease. 1. Structure of monosaccharides, general formula. The basic formula of monosaccharides is CH2ON, where N is typically 3 to 7, corresponding to the number of carbon atoms. For example, glucose, C6H12O6, has 6 carbon atoms. Carbon backbone. Monosaccharides consist of a carbon chain with hydroxyl groups, nice OH attached to most of the carbon atoms, and either an aldehyde nice CHO or ketone C equals O group. Aldoses and ketoses. Aldoses contain an aldehyde group, e.g. glucose. Ketoses contain a ketone group, e.g. fructose. Chirality. Monosaccharides often have multiple chiral centers, leading to different isomers, e.g. D and L forms. The D form is the most common in nature. Chamber 2. Classification of monosaccharides. Monosaccharides are classified based on the number of carbon atoms in their structure. Trioses, 3 carbon sugars. Examples include glyceraldehyde, aldotriose, and dihydroxyacetone, ketotriose. Important in glycolysis as intermediates. Tetroses, 4 carbon sugars. Examples include erythrose and threose, rare in nature but involved in certain metabolic pathways. Pentoses, 5 carbon sugars. Examples include ribose, aldopentose, and ribulose, ketopentose. Ribose is a key component of nucleic acids, RNA, and ribulose is involved in the Calvin cycle of photosynthesis. Hexoses, 6 carbon sugars. Examples include glucose, galactose, aldoses, and fructose, ketose. Glucose is a primary energy source, while galactose is found in dairy products and fructose is present in fruits and honey. Heptoses, 7 carbon sugars. Examples include cetoheptulose. They are less common and are typically intermediates in metabolic pathways like the pentose phosphate pathway. 3. Biological functions of monosaccharides. Monosaccharides play several key roles in living organisms. Primary energy source. Glucose is a central energy source for most organisms. It is broken down through glycolysis and further oxidized in the citric acid cycle to produce ATP. The brain and red blood cells, in particular, rely heavily on glucose for energy. Building blocks for larger molecules. Monosaccharides form the structural units of disaccharides, e.g. sucrose, lactose, and polysaccharides, e.g. starch, cellulose, glycogen. They also participate in the synthesis of nucleotides, which are essential for the formation of DNA and RNA. Precursor molecules. Ribose is an integral part of nucleotides like ATP, NADH, and coenzymes. Fructose is a substrate for glycolysis and is metabolized in the liver. Cellular signaling. Certain monosaccharides, such as glucose, play roles in cell signaling pathways that regulate processes like insulin release and cellular uptake of glucose. Monosaccharides are also components of glycoproteins and glycolipids on cell surfaces, which are involved in cell recognition and immune responses. Number 4. Digestion and absorption of monosaccharides. Monosaccharides are directly absorbed in the small intestine without the need for further breakdown, unlike more complex carbohydrates. Absorption process. Glucose and galactose are absorbed via active transport, SGLT1 transporter, while fructose is absorbed by facilitated diffusion, GLUT5 transporter. Once absorbed, these sugars are transported into the bloodstream and delivered to various tissues for energy production or storage. Hammer 5. Health implications of monosaccharides. Diabetes mellitus. Glucose metabolism is central to the management of diabetes. In diabetes, there is either insufficient insulin production or reduced cellular sensitivity to insulin, leading to high blood glucose levels. Monitoring blood glucose and adjusting dietary intake of simple sugars is critical for diabetes management. Fructose metabolism. While fructose is a natural sugar found in fruits, excessive intake, especially from high fructose corn syrup, has been linked to metabolic disorders like insulin resistance, obesity, and non-alcoholic fatty liver disease, NAFLD. Unlike glucose, fructose metabolism occurs mainly in the liver, where it can contribute to the synthesis of lipids if consumed in large amounts. Genetic disorders. Galactosemia, a rare genetic disorder that affects the metabolism of galactose, leading to its accumulation in the body. This condition requires dietary management to avoid serious complications. Hereditary fructose intolerance, HFI, a genetic disorder where the body lacks the enzyme to break down fructose, leading to toxic accumulation when fructose-containing foods are ingested. Hammer 6. Monosaccharides in nutrition and diet. Natural sources. Monosaccharides like glucose and fructose are naturally found in fruits, vegetables, and honey. Lactose, a disaccharide composed of glucose and galactose, is present in dairy products. Dietary balance. Consuming monosaccharides through whole foods, e.g., fruits, is preferred due to their fiber and nutrient content, which slow the release of sugars into the bloodstream. High intake of refined sugars can contribute to metabolic issues like obesity and cardiovascular diseases, making it crucial to manage simple sugar intake in the diet. Emerging research and applications. Monosaccharides in medical treatments. Research is exploring the role of monosaccharides in therapeutic interventions, such as the use of glucose for treating hypoglycemia. Certain sugar derivatives are being studied for their potential use in cancer therapy, as some cancer cells rely heavily on glucose for growth. Glycomics, the study of glycans, carbohydrate structures, and their biological roles, known as glycomics, is a growing field that provides insights into how monosaccharides and their derivatives function in cell signaling, immunity, and disease mechanisms. Understanding glycan structures could lead to advances in vaccine development and personalized medicine. 8. Conclusion. Monosaccharides are foundational molecules in biology, providing energy, forming structural components, and participating in complex biochemical processes. While they are essential for life, maintaining a balance in their intake is crucial for optimal health. With ongoing research into their roles and metabolism, the understanding of monosaccharides continues to expand, offering potential insights into their applications in medicine and nutrition. This review highlights the significance of monosaccharides in biological systems and their impact on health, underscoring their fundamental role as the building blocks of carbohydrates. Disaccharides, a review. Disaccharides are a type of carbohydrate composed of two monosaccharide units linked together by a glycosidic bond. They are an essential part of human nutrition and metabolism, providing energy and contributing to various biological functions. This review explores their structure, types, digestion, biological roles, and implications for health. 1. Structure of disaccharides. Composition. Disaccharides consist of two monosaccharides joined by a glycosidic bond, which is formed through a dehydration reaction, removal of a water molecule. The nature of the glycosidic linkage, A or B, influences the properties and digestibility of the disaccharide. Glycosidic bond. The bond can occur between different carbon atoms of the monosaccharides, e.g. C1 and C4 or C1 and C2. The most common types of glycosidic bonds are Ah 1-4 and Beta 1 to 4, referring to the carbon positions of the sugars involved in the bond. 2. Types of disaccharides. Disaccharides are classified based on the monosaccharides they contain and the type of glycosidic bond. Sucrose. Composition, glucose plus fructose. Glycosidic bond, AF1-2 linkage. Source, commonly known as table sugar. It is naturally found in sugarcane, sugar beets, fruits, and vegetables. Function. It is a major sweetener and provides a rapid source of energy due to the ease with which it is broken down into its monosaccharide components. Lactose. Composition. Glucose plus galactose. Glycosidic bond. Beta-1,4 linkage. Source. Known as milk sugar, lactose is present in milk and dairy products. Function. It is an important energy source for infants and plays a role in calcium absorption in the intestine. Maltose. Composition. Glucose plus glucose. Glycosidic bond OF1,4 linkage. Source. Found in malted grains. It is also a product of starch digestion. Function. It serves as an intermediate in the digestion of starch and glycogen, breaking down further into glucose for energy production. Trehalose. Composition. Glucose-glucose. Glycosidic bond OF1,1 linkage. Source. Found in fungi, algae, and some insects. Function. It acts as a source of energy and provides stability to proteins and membranes under conditions of stress, such as dehydration. 3. Digestion and absorption of disaccharides. Hydrolysis. The digestion of disaccharides occurs in the small intestine, where they are hydrolyzed into their monosaccharide components by specific enzymes. Sucrose is broken down by sucrase into glucose and fructose. Lactose is digested by lactase into glucose and galactose. Maltose is hydrolyzed by maltase into two glucose molecules. Absorption. The resulting monosaccharides are absorbed through the intestinal lining into the bloodstream, where they are transported to cells for energy production or stored as glycogen in the liver and muscles. 4. Biological functions of disaccharides. Disaccharides serve various biological functions in living organisms. Energy source. Disaccharides provide a quick and efficient source of energy due to their ability to be rapidly broken down into monosaccharides. The glucose derived from disaccharides is especially important for energy needs during physical activity and for brain function. Role in food. Sucrose is commonly used as a sweetener in processed foods, enhancing taste and palatability. Lactose in dairy products contributes to the nutritional value of milk, providing a significant energy source for infants. Maltose plays a role in the brewing and fermentation processes in the food industry. Prebiotic properties. Certain disaccharides may have prebiotic properties, supporting the growth of beneficial gut bacteria. For instance, lactulose, a synthetic disaccharide, is used to treat constipation and improve gut health. 5. Health implications of disaccharides. Lactose intolerance. Lactose intolerance occurs when the enzyme lactase is deficient, leading to difficulty in digesting lactose. This results in gastrointestinal symptoms like bloating, gas, and diarrhea upon consuming dairy products. Lactose intolerance is common in many populations and can be managed by consuming lactose-free dairy products or taking lactase supplements. Dental health. Sucrose is a primary contributor to dental caries, cavities, as it serves as a substrate for bacteria in the mouth, leading to acid production that erodes tooth enamel. Reducing sucrose intake and maintaining proper oral hygiene are essential for preventing tooth decay. Obesity and metabolic syndrome. Excessive consumption of sucrose and other added sugars is associated with an increased risk of obesity, insulin resistance, and metabolic syndrome. Monitoring the intake of added sugars, including those derived from sucrose, is important for maintaining metabolic health. Dietary considerations. For individuals with certain conditions like diabetes, managing the intake of disaccharides is crucial to maintaining stable blood glucose levels. Choosing complex carbohydrates over simple sugars can provide more sustained energy and reduce the risk of rapid blood sugar spikes. 6. Disaccharides in diet and nutrition. Natural sources. Sucrose is naturally present in fruits, vegetables, and honey. Lactose is found in milk and dairy products. Maltose can be found in products like malted cereals and beverages. Dietary guidelines. Recommendations emphasize limiting the intake of added sugars, including sucrose, to reduce the risk of chronic diseases. Consuming natural sources of sugars, like those found in whole fruits, is encouraged for their additional nutrient and fiber content. 7. Emerging research and applications. Disaccharides in biotechnology. Enzymes like sucrase and lactase are used in the food industry to produce various products. Lactase, for example, is used to manufacture lactose-free dairy products. Disaccharides are also used in biotechnological applications, including the production of biofuels through the fermentation of sugars. Therapeutic uses. Lactulose, a synthetic disaccharide, is used as a medication for treating constipation and hepatic encephalopathy. It works by drawing water into the bowel and reducing ammonia levels in the blood. Advances in understanding the digestion and metabolism of disaccharides could lead to new therapeutic approaches for managing metabolic disorders. Disaccharides and the gut microbiome. Research is exploring how disaccharides interact with gut bacteria, influencing the composition of the gut microbiome. The relationship between disaccharides and gut health is an area of interest, particularly in developing prebiotic therapies that support beneficial gut bacteria. 8. Conclusion. Disaccharides are crucial components of the diet and play key roles in providing energy and supporting various physiological functions. While they are essential for nutrition, their consumption needs to be balanced to prevent health issues such as obesity and diabetes. As research continues to explore the interactions between disaccharides and health, a deeper understanding of their roles in metabolism and disease management is emerging. This review highlights the importance of disaccharides in both nutrition and health, emphasizing their diverse functions and the need for mindful consumption in a balanced diet. Polysaccharides, a review. Polysaccharides are complex carbohydrates composed of long chains of monosaccharide units linked together by glycosidic bonds. They play a significant role in biological systems, functioning as energy reserves, structural components, and in cell signaling. This review provides an overview of their structure, classification, functions, digestion, and importance in health and industry. Number 1. Structure of polysaccharides. Basic composition. Polysaccharides consist of multiple monosaccharides, e.g. glucose, linked by glycosidic bonds. The arrangement and type of glycosidic bonds, or B, determine their structure and function. Linear versus branched. Polysaccharides can be linear. Monosaccharide units are linked in a straight chain, e.g. cellulose. Branched. Chains have branches where some monosaccharide units connect to multiple others, e.g. glycogen, starch. Homopolysaccharides versus heteropolysaccharides. Homopolysaccharides, composed of one type of monosaccharide, e.g. starch, cellulose. Heteropolysaccharides, composed of different types of monosaccharides, e.g. hyaluronic acid in connective tissues. 2. Types of polysaccharides. Polysaccharides are broadly classified into storage and structural polysaccharides based on their function. Storage polysaccharides serve as energy reserves in organisms and can be broken down when energy is required. Examples include starch, found in plants composed of amylose, linear, and amylopectin, branched. Amylose has OF1,4 bonds, making it a straight chain. Amylopectin has OF1,4 bonds with HOF1,6 branching, making it more compact. Major sources include grains, potatoes, and legumes. Glycogen, found in animals, highly branched with HOFA of 1,4 and alpha 1 to 6 bonds, stored mainly in the liver and muscles, providing a quick source of glucose when needed. Its structure allows rapid release of glucose during periods of high energy demand. Structural polysaccharides provide support and structure to cells and tissues. Examples include cellulose, composed of Bet1 to 4 linked glucose molecules forming straight chains, found in plant cell walls, providing rigidity and strength. Humans lack the enzyme cellulase, making cellulose indigestible and a source of dietary fiber. Chitin, composed of N-acetylglucosamine units linked by BCA1,4 bonds, found in the exoskeletons of insects, crustaceans, and in the cell walls of fungi, provides structural strength and protection. 3. Digestion and absorption of polysaccharides. Hydrolysis by enzymes. Polysaccharides must be broken down into monosaccharides for absorption. Starch digestion begins in the mouth with salivary amylase and continues in the small intestine with pancreatic amylase, breaking down starch into maltose, maltotriose, and dextrins. Cellulose digestion. Humans cannot digest cellulose due to the lack of cellulase, making it an important source of insoluble dietary fiber. Absorption. The resulting monosaccharides, e.g. glucose, are absorbed in the small intestine and enter the bloodstream, providing energy to cells. 4. Biological functions of polysaccharides. Polysaccharides play numerous roles in living organisms. Energy storage. Glycogen serves as a readily available energy reserve in animals. It is broken down during fasting or exercise to maintain blood glucose levels. Starch is a primary energy source in the human diet, providing sustained energy through its slow digestion. Structural integrity. Cellulose is a key component of plant cell walls, maintaining their structure and helping plants remain upright. Chitin contributes to the strength and protection of fungal cell walls and insect exoskeletons. Protective roles. Mucopolysaccharides, glycosaminoglycans like hyaluronic acid, play crucial roles in lubricating joints, maintaining skin hydration, and providing structural support to connective tissues. Heparin, a highly sulfated glycosaminoglycan, acts as an anticoagulant in the blood. Cell communication and immune function. Polysaccharides on cell surfaces are involved in cell recognition and signaling. For example, glycoproteins and glycolipids with polysaccharide chains play roles in immune responses. Health implications of polysaccharides. Dietary fiber. Polysaccharides like cellulose, hemicellulose, and pectin form the bulk of dietary fiber. Insoluble fiber, e.g., cellulose, aids in digestion by adding bulk to stool and promoting regular bowel movements. Soluble fiber, e.g., pectin, beta-glucan, helps lower blood cholesterol and regulate blood sugar levels by slowing digestion. A diet rich in fiber is associated with reduced risk of heart disease, type 2 diabetes, and certain cancers. Metabolic health. Complex polysaccharides, such as starch, have a lower glycemic index compared to simple sugars, leading to a more gradual release of glucose into the bloodstream. Resistant starch, a type of starch that resists digestion, can have prebiotic effects, supporting the growth of beneficial gut bacteria. Autoimmune and inflammatory conditions. Some polysaccharides have immunomodulatory properties, influencing the immune response. Beta-glucans, found in oats and fungi, have been studied for their ability to modulate immune function and may support the immune system in fighting infections and cancers. 6. Polysaccharides in industry and biotechnology. Food industry. Polysaccharides like starch, pectin, and alginate are used as thickeners, stabilizers, and gelling agents in processed foods. Modified starches are utilized to improve the texture and stability of various food products. Pharmaceuticals and medicine. Hyaluronic acid is used in skincare products for its hydrating properties and in joint injections for osteoarthritis treatment. Heparin is a critical anticoagulant drug used to prevent blood clotting during surgeries and in treating certain cardiovascular conditions. Chitosan, derived from chitin, is used in wound dressings for its biocompatibility and antibacterial properties. Bioplastics and biofuels. Polysaccharides like cellulose and starch are explored for their potential in producing biodegradable plastics and as raw materials in biofuel production. The development of cellulose-based biofuels aims to provide a renewable and sustainable energy source. 7. Emerging research and applications. Prebiotics and gut health. Research into polysaccharides as prebiotics focuses on their role in supporting gut microbiota. Non-digestible polysaccharides like inulin can stimulate the growth of beneficial bacteria such as bifidobacterium. This area is promising for improving digestive health, immune function, and even mental health through the gut-brain axis. Polysaccharides in drug delivery. Polysaccharides like chitosan and alginate are being studied for their potential in drug delivery systems due to their biocompatibility, low toxicity, and ability to form gels or films that can encapsulate drugs for controlled release. Regenerative medicine. Polysaccharides like hyaluronic acid and chitosan are investigated for their role in tissue engineering and regenerative medicine. They can be used as scaffolds for cell growth and tissue repair due to their ability to mimic the extracellular matrix. 8. Conclusion. Polysaccharides are diverse and complex carbohydrates that serve critical roles in biology, from providing energy to forming the structural basis of plants and other organisms. They are integral to human health, offering dietary benefits such as improved digestion and metabolic regulation. In industry, their applications extend into food technology, pharmaceuticals, and renewable materials. As research continues to uncover new uses and benefits, the importance of polysaccharides in science, medicine, and nutrition remains profound. This review underscores the multifunctional nature of polysaccharides and their relevance in both natural and applied contexts, highlighting their significance in sustaining life and advancing technology. Proteoglycans and glycoproteins are both types of glycosylated proteins, meaning they have carbohydrate chains attached to their protein backbone. Despite sharing this similarity, they differ significantly in structure, composition, and function. Here's a comparison between the two. 1. Structure and composition. Proteoglycans consist of a core protein to which one or more glycosaminoglycan GAG chains are covalently attached. Glycosaminoglycans, GAGs, are long, unbranched polysaccharides composed of repeating disaccharide units that often include amino sugars like glucosamine or galactosamine and uronic acids like glucuronic acid. The carbohydrate component forms the majority of the proteoglycan's mass, up to 95%, making proteoglycans highly hydrated and often large in size. Examples of GAGs include hyaluronic acid, chondroitin sulfate, and heparan sulfate. Glycoproteins, composed of a protein backbone with shorter, branched oligosaccharide chains attached. These carbohydrate chains can vary in length and composition and are usually attached through N-glycosidic or O-glycosidic bonds. The carbohydrate content in glycoproteins is generally lower compared to proteoglycans, often 1 to 20% of the total weight. The sugars found in glycoproteins include mannose, fucose, galactose, and sialic acid. 2. Functions. Proteoglycans primarily serve structural and mechanical functions in connective tissues. They are major components of the extracellular matrix, ECM, and contribute to the formation of cartilage, providing it with resilience and the ability to withstand compressive forces. Due to their highly negative charge from sulfate and carboxyl groups in GAGs, proteoglycans attract water, forming a gel-like structure that helps with tissue hydration and shock absorption. They are also involved in signaling processes by interacting with growth factors and other molecules in the ECM. Glycoproteins have diverse functions, including cell-cell recognition, adhesion, and immune responses. They play a role in the structure of cell membranes, forming part of the glycocalyx, a carbohydrate-rich layer on the cell surface. Glycoproteins are essential in immune system functions, as they include antibodies, immunoglobulins, cell surface receptors, and signaling molecules. Many hormones, such as erythropoietin and certain enzymes, are glycoproteins, which helps with their stability and proper function. 3. Location. Proteoglycans, predominantly found in the extracellular matrix, ECM of connective tissues like cartilage, tendons, and skin. They are also present on the cell surface, where they can interact with receptors and other molecules. Examples include aggrecan in cartilage and perlecan in basement membranes. Glycoproteins, found on the cell surface, in the extracellular matrix, and as circulating proteins in blood and body fluids. They are a part of cell membranes, where they play a role in cellular recognition, such as MHC, major histocompatibility complex, proteins involved in immune response. Examples include antibodies, immunoglobulins, glycophorin in red blood cell membranes, and transferrin, which is involved in iron transport. Part 4. Carbohydrate attachment. Proteoglycans. The carbohydrate chains are long and consist mainly of GAGs, which are usually attached to the core protein through a serine residue via a special linkage, e.g. tetrasaccharide bridge. The GAGs chains are often highly sulfated, contributing to the negative charge and ability to bind water. Glycoproteins. The carbohydrate chains are shorter, branched, and more diverse, often attached to the protein through N-linked, asparagine or O-linked serine or threonine glycosylation. These carbohydrates are less structurally rigid than those found in proteoglycans and do not have the same extensive hydration properties. Summary table. Phe proteoglycan, glycoprotein, glyprotein. Carbohydrate-type long chains of GAGs, e.g. chondroitin sulfate, heparan sulfate. Short branched oligosaccharides, e.g. mannose, sialic, carbohydrate proportion. Up to 95% of total weight, 1 to 20% of total weight. Main function, structural support, hydration, and shock absorption in ECM, cell recognition, signaling, immune response, location, extracellular matrix, cartilage, connective tissues, cell surfaces, blood, body fluids, and membranes, attachment to protein via a tetrasaccharide linker to serine residues, N-linked asparagine or O-linked serine threonine. Conclusion. Proteoglycans and glycoproteins, though both glycosylated proteins, serve different structural and functional roles in the body. Proteoglycans are primarily associated with the structural integrity and hydration of tissues like cartilage, while glycoproteins play crucial roles in cellular recognition, immune function, and signaling. Understanding the differences between them is essential for studying their roles in health, disease, and therapeutic applications.